And I would like to introduce our speaker, and she's in the room with me.
Hi, everyone. Good afternoon. My name is Dara Reynolds, and today I'm going to be presenting on the invisible workers in the soil. I have a background in biology. I got a bachelor's in biology in 2015 from WSU Vancouver, and I graduated as a master gardener in 2021. I also have about 10 years of gardening experience. I definitely have grown along my gardening journey. I have started with adding a lot of chemical fertilizers and tilling a lot, and slowly I have started to add more organic compounds and no tilling methods. So with that, I'll go ahead and get started. Can they see my?
Yep,
Did they see that or they see this?
they see that.
Okay. Not everyone realizes, but the soil below our feet is beaming with life. Soil is a valuable resource. When healthy soil has bacteria, fungi, and other microbes living in a symbiotic relationship,
That was me. I hit something. There you go. You're okay.
okay. There's a fun fact, the number of soil organisms in just 1 tablespoon of healthy soil can be more than 9 billion. This is because the cell size of microorganisms are so small. If you look here at the picture of a root, you can see just how much surface area a root can have. Now imagine that with all the small cells, that in this area you have tens of billions of microbes. The roots exude microbial food to attract and feed microbes that provide plants with the nutrition they need. Today we will discuss how the organisms that are in soil, the soil health and benefits, the nitrogen, potassium, and phosphorus solubility, that is what makes them usable in the soil, possibility of using cover crops and non-till methods, soil testing both at home and send out, and ways to amend your soil. As you look at this chart over here, you can see organic matter, and you have things like fungus and bacteria, protozoa, and nematodes. The ones that we know a lot about are the bacteria and fungus, but we also are going to touch on the protozoa, and then also we have the nematodes that we'll touch on the end, and as well as the arthropods that are not microorganisms. The basic food web includes microbes. The microbes we know the most. Oh, sorry. We will also talk about prokaryotes, archaea, and even viruses as part of soil health. The rhizosphere is the area of soil that is influenced by root secretions and soil microorganisms. It is the area of most concentration microbial activity that is right below the surface. This is here the general nutrient and water cycling occurs. So bacteria. Bacteria is about 1 micrometer in size. They are the big guys in the nutrient cycling. There is about 60,000 different types of bacteria that are known at this time, but there's probably even more. They break down organic matter and are part of the nitrogen, phosphorus, and sulfur cycles. They exude chemicals by the roots and sometimes form root nodules where close to the root and in the root more breakdown can occur. Actinomycetes are a kind of bacteria that break down organic material that are complex and difficult for other organisms to break down. Examples include cellulose, lignin, and chitin. This forms the humus, or the dark organic matter that excuse me. It forms a dark organic matter that is part of the soil and makes up the non-living part of the soil. It is what gives the earthy smell. Now there's fungus. Here I did a picture of some of some mycelium in the soil.
Mm.
You can see the little white hairs. They can also form mycorrhizae, which go deeper into the soil, and they create transport for nutrients from the roots of the soil into deeper. And they also help roots get water that they themselves cannot reach. The decomposers are called saprophytic, and the ones again that transport the nutrients are called the mycorrhizal. They are the structure builders.
What did I do? Okay.
And in exchange for carbohydrates, mycorrhizal fungi extend reach, accessing nutrients and water. Plant roots. The fungal hyphae increase soil structure by binding soil particles and forming aggregates. This improves structure, water retention, and aeration. Some are soil-borne pathogens, and some are pathogenic to arthropods and nematodes. And they also can kill soil-borne pathogens as well. The hyphae are what form into larger mycelium networks. These mycelium networks are known as some of the biggest organisms in the world. So you have a very small organism that makes up a network that all communicates, that is considered one of the largest organisms in the world. I'm not switching. Oh, it is.
Okay. Here.
Oh, it's the picture here, and then it will go to the next slide.
Right here.
Are we able to go back to this? Are they seeing this or this?
That way.
Are you guys able to see the slide?
Everybody's.
Yes, we can see the slides.
Okay.
Zoa?
Yes, thank you. Okay. So the protozoa, they are single-celled organisms that mostly eat bacteria. They release nitrogen as they eat the bacteria. They also eat protozoa, soluble organic matter, and sometimes fungi. The two main types of protozoa are ciliates and flagellates. There are less ciliates, but they are larger, and they can eat up to 10,000 bacteria in a single day. The ciliates have cilia around them, and they move around with an oar-like motion, while the flagellates have a flagella, and they kind of just bounce around and bounce into things. It's a very characteristic things of flagellates. Both of these help regulate the microbe population and break down things for the nutrient cycle. Now we have archaea. Archaea is similar to bacteria, but genetically different. They are a main part in ammonia oxidation as part of the nitrogen cycle. Here you can see that you have the ammonia breakdown, and you have the phosphorus breakdown, the carbon breakdown, and even silicon. There's still not much known about silicon and what it does to benefit soil, but we do know that archaea does play a part in breaking down the silicon. Archaea is one of the organisms that we really don't know that much about and exactly how it does the chemical reactions, but we are learning more and more about them. Archaea are more prominent in extreme climates, and that's where they're originally found. And so in an area where there's more extreme climate, you're going to have many more archaea than you are going to have in, say, a forest that doesn't have the extreme climate that a glacier area would have. But archaea are around in extreme environments, and they may increase when you have things like drought and flood. So viruses are actually very prominent in the soil, and they're actually sometimes there's more viruses than there are bacteria. Viruses are even smaller than bacteria. They're really only made up of amino acids and protein. These type of viruses are called phages. They do not infect the plant. They're specifically attacking bacteria and archaea. Another name for them is bacteriophages. They're important for regulation of bacteria populations. They influence nutrition cycles, and they have the potential to suppress plant pathogens. They can outnumber bacteria, and in a desert, there's about 10 to the third power in one dry gram of soil, while in a forest, there's about 10 to the ninth power in one dry gram of soil. Viruses that kill the bacteria are called lytic viruses, while viruses that integrate into the bacteria's genome are called temperate viruses. And when I say that they incorporate into the genome, that means that they actually go into the gene of the bacteria, and there's a gene transfer that occurs. Again, there's many viruses that are like archaea that are largely not identified. There are so numerous amount and so small that they are difficult to find, difficult to measure, difficult to control in a lab-like setting. And nematodes. Nematodes largely benefit agriculture. There are pathogenic nematodes. With that being said, nematodes also eat other nematodes that are pathogenic. So if you have the good guys, you're helping keep away the bad guys. So one thing they do is they eat plant parasites, and then they also break down they mineralize nutrients into plant available forms. It's thought that 8 to 19% of nitrogen mineral comes from nematodes. There are food sources for other soil organisms such as insects, centipedes, mites, fungus, and earthworms. They spread bacteria and fungus as they graze through the soil. So an area that does not have the bacteria needed or the fungi needed may be inoculated by nematodes as they spread. And then there's the visible workers, the invertebrates. You have the worms, the insects, the millipedes, the mollusks, which are the snails and the slugs, and bees. There's also many more organisms that contribute to the soil ecosystem. All of these organisms help break down, they help with aeration, and they also help with pollination. Specifically, the earthworms help with aeration as they go through the soil and they make holes and they allow farther for water and air to reach into the soil. So here we're going to touch on the different cycles. The main cycle that people have probably heard about is the nitrogen fixation cycle. Or the nitrogen cycle. As the first part of the nitrogen cycle, you have the nitrogen fixation. Here, some bacteria can convert atmospheric nitrogen into usable ammonia. So the bacteria get it from above ground in the air and they create it into a usable form. And then there's nitrification. A different type of bacteria can convert the ammonia into nitrite and then nitrates, making nitrogen readily absorbed by plants. After this is done, it can also go back out into the air called denitrification. This is a conversion by bacteria of nitrates back into the atmospheric nitrogen. So the cycle will continue. Then there's the phosphate. Phosphate is often not in a usable form and has to be solubilized into a usable form. This is largely done through the release of organic acids. Phosphorus then impacts energy storage transformation, cell structure, respiration and photosynthesis, cell enlargement and division, and root development. Then you have the potassium solubilization. Potassium solubilization microorganisms are also referred to as KSM. Potassium in the element table is actually used, you have the K. So don't be confused with the phosphorus right here, which it goes by P. Potassium is the letter K to represent. It uses the letter K to represent. Organic acids reduce soil pH and dissolve potassium-containing molecules. So again, it is also done just like the phosphorus. You have the organic acids that break down the phosphorus to make it soluble, and then you have the organic acids that break down potassium to make it soluble. Also, another thing that is done in the solubilization of potassium is when bacteria forms biofilms on potassium-containing minerals. Potassium is important in plant growth and mineral yields. And then it also, when you have microbes that are breaking down potassium, it decreases the cost, as potassium is one of the more expensive nutrients to add to the soil. Having usable potassium decreases pathogenic microbes, and then it also decreases heavy metal buildup, as non-used potassium can sit in the or non-usable potassium can sit in the soil and actually create a metal buildup. So here we're going to talk about soil health. Soil health is the capacity to sustain plants and animals and humans. Carbon, nitrogen, phosphorus, and other nutrients can be stored, transformed, and cycled as part of the soil food web, as we have just talked about. This provides physical stability and support. The tilth, or the structure of the soil, can support root growth and allow water movement when soil is healthy. As soil increases in function, soil is able to absorb and retain more water, making less runoff and erosion. This leaves water accessible for plants and creates more drought and flood resistance. So say you have soil that is less compact, you're going to have, when the water runs over it, you're going to have nutrients that are going to run out. You're going to have soil that's going to run out, and then you're not really going to keep around what you're adding to your soil. When if you have soil health working with you, you're able to keep what you need in your soil more without as much runoff or land erosion in general. More and more farmers are talking about increasing soil health. One option is the no-till method. You can, as part of the no-till method, you can use cover crops. You can do diverse rotations, including having livestock grazing in the areas. And you can also increase organic matter. This in turn increases and diversifies microbial activity, reduces soil compaction, and improves nutrient and storage cycling. So tilling. Tillage, especially during the warmer months, reduces water infiltration and increases runoff. This makes the soil more erosion prone and decreases biological cycles. Benefits to not tilling is you get more organic matter and moisture for plant growth, greater carbon retention and cycling efficiency, reduced soil erosion, decreased costs of adding nutrients to maintain yields. Cover crops also allow food and escape for wildlife. Proper grazing allows pasture plants to rest and regrow. So cover crops. Some common cover crops are rye, wheat, oats, clover, legumes, turnips, radishes, and triticale. Here in my picture right here, I am actually using a coastal strawberry as a cover crop. As you can see there, you have one dandelion weed coming out, and then you have some branches here or some sticks here from crops from last year. I often leave old debris in my garden, as it will naturally decompose, and it will also allow an environment for organisms around, and leaving the roots in the ground allows the life to continue and allows aeration to continue as well. So the cover crops is a way of naturally adding the organic material and like I said, improving water filtration and the weed suppression. When the cover crop is done, to put in your new crop without tilling, you can mow. Some of the crops naturally die off. You can cover for small farms, you can cover things with the tarp. You can actually plant within some cover crops, as they don't compete very much. Or a cover crop with, can be, or you can cover with additional organic material that is able to suppress the cover crop. So soil testing. Evaluating your soil drainage. Are we able to move this? I can't see the full.
No, people don't see this.
No, I can't see the full slide.
Oh, sorry. There
Okay,
you go.
thank you. In a damp area, so this is a soil test that you can do at home. I don't know how many of you guys have heard about this before. This is one that I learned as a master gardener myself. So you start by digging a 6-inch deep hole that will fit a 46-inch tin can. You remove the bottom and the top of the can and you place it in the hole. Then you go around the can and firmly surround it with dirt. Next, you fill that can with water. In an hour, it should drop 2 to 3 inches if your soil is where you want it to be. If it drains too fast, it can decrease. You can decrease that drainage by adding organic material such as compost and then cover with mulch. If it drains too slow, you can aerate and or add sand, perlite, add drainage or mulch. Here's another way to test your soil at home. It's by separating soil components. You would dig a 6 inches deep into the soil and loosen up the dirt. You would remove any stones, any soil clumps, and any roots, and you fill a jar about halfway full and wet in the soil until its mud-like consistency is formed. You settle the soil in the jar by tapping it onto a flat surface until it's compact and marking the level. You fill the jar then with water to the top and add a tablespoon of dishwashing detergent. This helps separate the layers. You then place the lid on the jar and shake for several minutes until it is completely mixed. Then you let the jar settle on a flat surface. 3 layers from this will form. You'll have the sand, the silt, and the clay. The sand will separate first very quickly, and it will form on the bottom. The silt will form in the middle, take an hour or so, and then the clay will take a while to form, and it will form on the top. You should be able to see defined lines of each. You should have about an even mixture of each of these things in order to have the one in soil. So adding organic matter. Some ways you can add organic matter is by making your own compost pile. You can add aged manure. You can add mulch. Mulch can include woody byproducts such as mulch bark, sawdust, and wood ash. You can add plant residue, lawn clippings, straw, and leaves. You can add bone meal or even biosolids. Here is a picture of some garlic I have, and what I have underneath is I have some compost underneath, and with some manure, and then I also have straw over top. This helps create a weed barrier, and it makes it to where I don't have to do a whole lot during the off season. And then I also have some more crops growing here in the back as well. So then there's the chemical component testing. In general, most vegetable crops want a pH from 6 to 7.5. It's rather basic. But then there's the acid-loving plants. You have it. The lower pH is going to be more acidic, which is a 4.5 to a 5.5. So higher pH of the alkaline. In order to decrease, you want to add some sulfur. If you have a low pH, as in it's too acidic, you can add lime. So when you do soil testing, the different nutrients you're gonna, this is all gonna show in the soil testing, but the nutrients that are needed, but that not all of them are tested for, are as follows: the nitrogen, phosphorus, calcium, magnesium, sulfur, boron, iron, magnesium, zinc, copper, fluoride, the molybdenum, and the nickel. Then you also have nutrients that are sequestered from the air and water, the carbon, hydrogen, and oxygen. As for the lab soil testing, you have the phosphorus, potassium, calcium, magnesium, boron, and the pH that is shown when you send soil out. That is specific in Western Washington. In places like Eastern Washington, they're not gonna give you the lime requirements. They're gonna probably talk more about adding the acidity instead. In order to do a soil test, you want to define a specific area that has been treated the same way that's going to have similar samples throughout. You want to take 10 samples that are 6 to 10 inches deep, and it is ideal that you're using a soil probe. You can find those online. They're about $20. After you take your samples, you want to put them all together and mix them. You let them air dry and then mix again, and then you'll send off in a labeled bag. And then with that label, you want to include where you got your samples. Here are some sources I have. And then any questions?